And I want now to go to a slide that is not here coming. I need to go through it. Sorry guys. Yeah, this one. I like it. Okay, so here is why you're a bioinformatician. Because of more of an inability than an ability. I'm sorry to tell you, but that's how life can be sometimes. So here, this is the physicist world. This is fantastic. You know, suppose we were there already. You give me a DNA sequence. I perfectly have to say, okay, here's one gene, there's the other gene already. That's difficult, right? To predict where genes are. Start codon, stop codon, data box. This is for the binding sites. So you have all the genes. Then you translate the protein just in a computer, right? On a piece of paper, but that's a lot of work. The codon table, you have the protein sequence, and now you say, okay, now I have another computer program that will just calculate, or it knows the physical rules, it knows exactly how this protein folds into a hemoglobin structure. And now we have this structure, and now we know here is the active site, so this protein is doing this. And I know exactly what, how the reactions are going to happen. So this is what we would like to do. Given a sequence, we would like to be able to, for example, predict the protein structure. And when we have that, we would like to be able to say, okay, given this structure, this is an alcohol dehydrogenase, or this is hemoglobin. This is a protein that transports oxygen at this rate. A horse can run faster than humans because the hemoglobin is a bit optimized. So we can all do this. That would be fantastic. We could cure a lot of diseases. The problem is, you can see the color of this arrow, red light, not possible. We can't do this. A little bit. Techniques are getting better, but this is in principle not done. So, when we have a sequence, we don't know how to do this, so we're stuck, right? How do we predict the function? We can't, not on first principles, not forward. And then a bunch of people got so frustrated. They said, but hold on, is there hope? I don't know. People have worked tremendously hard in biology and in the life science. They've measured a hell of a lot of things. There are many proteins. They've done experiments finding out what the protein is doing. All kinds of mutation experiments and knockout experiments, right? You find them, and they typed up the information in databases. Fantastic. Can we use that? And that's the idea now. You know what? We don't know the physical principles, but suppose we have in that database there is one protein. We know the structure of, because that's been measured, cumbersome techniques, and we know the function. And now we have another species and there is a protein sequence in, and we have that protein sequence, but we know nothing about it. But now you find out, hey, I compare this protein to that guy where I know something about. Hey, these two sequences are similar. Do I dare to guess that this similarity might be due to homology? If so, I can guess that means then these two guys, once upon a time, how long ago, no clue, but they were the same thing. So could it be that given that I know one thing for sure, then they were doing the same thing, right? What they do now, we don't, but the guesswork is, and that's what we're going to do is, if similarity is enough, it will be homologous, and then we just postulate, if you like, they will do the same thing. And you know what? 98% of the cases, this works. So, not knowing the physical principles, we're going to reason back. We can't go forward, we do it backwards, right? We do it the other way around. So what do? We have an unknown gene. We compare it against a database of stuff where we know about. If we find similar sequences to the level that we say, let's infer homology here, a putative homolog, that's what we call that. It's a likely homolog or a putative homolog, it's terminology. And it is copy all we know. And that works, it's that simple. Very often that works. And there is a whole program, some of you might know the name of the program already, it's the most widely used program in the life science. It runs millions of times per day. Complete people in big pharma have jobs on running this program called BLAST every day. If any of you would like a job like that, but I certainly did jobs. But anyway, so we're going to do this. So this is why you're here in this course. You're going to do this. You have to understand homology, paralogy, to understand how I call that a 4 o'clock moment, right? Okay, quickly, I'll fill you in. This is the basic principle. I could send you all home now, but I won't, because there are a few more slides. And I will go, having told this. So, basically this is what I just told you. You compare with something you hopefully know stuff about, right? And if sequences show enough similarity, we can be bold enough to assume in English, homology, okay? So this is the homology principle. So if you think there is a homologous relationship, that makes it more likely that they're doing the same thing, because once upon a time they were the same. So So it's nice, homology is a binary property, I told you that, right? And it's a nice, so here you go, when an unknown gene acts as homologous to a known gene, a gene where you know stuff about, you can just transfer that information onto X. So basically the picture is this. Unknown sequence, known sequence, where you know stuff about, you infer homology by similarity, comparing, and then you say, okay, all I know about this. Whoop. So you go to that so a biologist comes in your room and says, look, I have sequenced this bit of DNA and I made it into a protein, for example, and you know, now I want to do a lot of lab work on this, it will take me 5 years. I don't want to work on something that's rubbish, not important. Tell me, should I work on this? This is also part of the question. I'll stop certainly. You say, oh, of course, I'm a bioinformatician. I've done this course, Fundamentals of Bioinformatics. I will blast for you, right? I'll tell you the function. Come back tomorrow. This program is fast. Come back in an hour. The program is even faster. Okay. So the biologist comes back and you say, you know what? Nice to hear. I've done blast. Good news is I found the sequence. Also good news is, and it was a human sequence this biologist came in, right? You know, blast, I checked the protein, this is a similar protein, I think it's homologous, right? And you know what? It's a bacterial protein, I know that. But the function is not in the database. What can you do? What's your message to the biologist? Do you say, sorry, bye? Or do you advise this? What was the question of the biologist? Should I start working on this or not? What is your advice? What do you think? What did you establish? A relationship, a homologous relationship between a human gene and a bacterial gene. Now suppose, what is an important gene? Something that is, you know, for breathing oxygen. What is maybe a less important gene? You know, for man, there is a single gene that encodes hair on your ears or not, right? I know it makes it more attractive or not, but anyway, it's maybe a less essential gene than some of the genes that allow you to run away, for example. Right? So what's your advice? I know one thing for sure, if you agree with me, bacteria do not have hairs on their ears, right? Meaning the set of genes they have is probably pretty basic and important housekeeping genes. So your advice, biologist, even in not knowing the function is, given bacteria, human, go work on this because this is an essential gene. I'm sure about it. Not knowing the function. And this body walks away shaking. How can they say this? They say they don't know it, and they can still tell me to work on it. Crazy guys. That's how we are. Okay. Okay. So anyway, so here we go. Pretty quickly, homologous relationship. CA from now on in your book is common ancestor. Yeah? And so we have derived sequences. Europe, America, say they became pretty different. But why does it still work? There is one golden rule here. That's very nice that is happening. We observed, just observing. We looked at different genes, at different protein structures, and we said, hey, these genes are similar. And oh, and this is a pretty different sequence. And then you often find hemoglobin, for example. Very important protein. Between dogs, mice, human, horse, they can be 90% different, meaning 9 out of 10 letters will be different. Still, their shape is the same, pretty similar. And that allows us to say structure is more conserved than sequence. Sequences can become very different, but still the underlying or the encoded structure will be much the same. And that means it will be awfully good if you can all the time compare at the structural level, because that is where more similarity is. But the problem is we don't have as many data. Sequence data will. So we do it at the sequence. Anyway, this is the story. Sequences can be very different under divergent evolution. Still, the structures are maybe the same because the structure is the same, and if the active site has the same amino acids, then the function will be the same. And this allows us to play this blast track. If this would not be true, or if this would read sequence more conserved than structure, forget blast. Cannot happen. Can't reason like that, right? But now it works, so that's very nice. Okay, quickly on to now we start filling in. Now you're going to tell me what happened to evolution. You start guessing as an evolution biologist, okay? Here's a common ancestor. This is a protein, very short protein. Eh? Proteins are 300 to the champion of the world is nearly 30,000 amino acids. It's called titin. The name implies its size. But anyway, Here is a short protein, 4 letters. Now, we were there when evolution happened. So this protein, what happened? The letter B in the ancestral sequence was changed into C. You see that here, this B here became C then. Here something else happened. Do you see what happened? The C dropped out. We call that a deletion. That happens all the time in evolution. Big chunks can drop out or come in. So this is how you can encode it. C is down the drain, out, taken out. Deletion. Now, what is the problem? We would like to, now the question for me to you is, are these two sequences homologous? You know they are, because we know this process. They derive from a common ancestor. But normally, you have these two sequences, the common ancestor is not there. You have to start telling me based on what? Whether these sequences are enough similar that you would say, I dare to guess this is homologous. Now, here we go. We start doing alignment now. This is lining up the two sequences here. It's in your way, so I'll go back. I'm going to use the pointer here. So this is the one sequence here, and this is the other one. If we align them, how would the reasoning be? It's just short, we don't need a program. You see two letters that remain the same, the A and D, and you say, you know what? Seeing this letter, my assumption is this A was in the ancestral sequence, an A, and this D was D in the ancestral sequence. Nothing happened to this letter. No mistake. You're even right if you say that. Now you have a problem, because then you need to align a B to either this C or that C. Is there any reason to say this over that one? If you look at the alignment, it's called an alignment. This is hard. But now knowing what happened, what is the real situation? Which alignment is true? The right one or the left one? The computer will tell you if you're slow. Time silently till 5, and then I'll show you. Anybody? Left or right? Start yelling, guys. I hear left more than right. Right? Left? There's a whole movie called Clockwise on this computer. Anyway. Yeah, the leftish won once again. Very good. Democrats over Republicans. Anyway. Okay. So, so this too, because that letter was mutated in the first C, right? But you can imagine if you now have sequences of 500 long, a whole bunch of them, we need to solve this, align according to the evolution scenario, hopefully. That's a big job. It's not easy. It's a lot of guesswork. Here you have alignments. Train your eyesight. What do you see? You're good enough. What do you see? This is a boring alignment, right? DNA is only 4 letters. This might happen. What does this mean? I can tell you this is not a letter. Well, no, it's not a letter even, but it's certainly not one of the bases of DNA. This is just a placeholder to say CGT. What? Something happened here. In evolution, CGT came in this sequence or dropped out in the other. You can't tell if you don't know what happened, whether there is an insertion or a deletion, okay? So this is one alignment, this is another alignment. Do you see a nice motif here? Imitation. Who sees a motif here? Read. Some people have connected the protein sequence database to the Oxford Dictionary, and they try and find the longest English word in protein sequences, which can happen because you have 20 letters. And the word champagne, you can make it 10. Anyway, this is not a biological motif, I can tell you. Anyway, I should draw to a close. I will go a few minutes over time, but I want to show you this one to give you further feel, putting in a bit of structure. So this is an alignment of 9. This is 9, you count. Multiple alignment this is, more than 2 sequences aligned of sequences that are all homologous. Each of those sequences, structure more conservative sequence, falls into this structure. This is the 3-dimensional structure of each of those protein sequences. Now we do an experiment, given these positions, some positions, and I'm not allowed, you don't need to read it, but some letters are totally conserved, the same letter in one column, and this goes back to the ancestral sequence. So conservation, no mutation, and some other positions here, for example, you see a lot happen. They started mutating away. Let's check on this. What's the importance of these positions in the alignment of each of the letters in the ancestral sequence? We look at the structure. And here they did an experiment here. So there is a relationship between an alignment, the sequences, and the structure. What did people do? Here we got this idea of one protein called isocitrate dehydrogenase. Homology term. What happened here? They checked which amino acids remained the same. They're bluish colored here on the structure. And which amino acids, according to that alignment I showed you a moment ago, have changed. That's red, and white is in between. You see these yellow dots? This is the front side of the protein, this is the back side. Just a picture of two the same protein from either side. This is the active site. These bits here, the yellow bit, and these is the important position where the protein is doing what it should be doing. What do you see? Where is it more conserved? Where is it more blue? Do you agree with me that closer to the yellow dots there is more blue than you? Now we have established the important bit. What is important? Think about Darwin, how you feel about it, is conserved. If something is important for the function, in the active site there is this amino acid. Mutate it, it won't do it anymore. You disrupt your protein. So that's an important principle. I already alluded this. So in the rest of the course, we're going to do, using this program last, we're doing the practicals, this will be a practical, and you're going to run through a database with more than, at the moment, is growing by the week. Roughly 7 million annotated sequences, meaning stuff is known about it and that has been put by these citizen biologists in the database. So you run this. This I showed you. On databases, computers grow exponentially, the power doubling per 1.5 years, okay? Some sequence databases, the doubling time is 6 months. We are measuring, and what does that mean physically? So 1.5 years later there is a double this space, say, but this grows with the doubling time of 3 times shorter. And that means one exponential curve is like this, but this goes like that. So physically we will run into problems storing bio stuff alone, right? Think about it. We need you to solve this problem. And this is an exponential curve. Straight line means exponential growth, and sometimes. So what is happening? This is the last point, and then I send you out. Apologies for 2 minutes late. Mind the gap that you can find in the tube in London, but here the gap is between measuring. So sequencing is cheap nowadays. The Human Genome Project, the first human, cost roughly $3 billion. Not too bad actually for the first time, but okay. Now you do it well. The cost come down. It was $1,000. It's now $100. It will be 30, 40, or even less. Cheaper than your passport. Right? Think about it. So, but measuring protein structures or certainly function, you need to do all kinds of mutation experiments. Takes an awful lot of time. So we have far more sequence data than structural and functional data. Mind the gap, and the gap is growing. So there is an ever-increasing need to reason with sequence data, predict everything you can find, because we need to understand the biosystem, right? That's this course. We'll work on it with the practicals. Okay. A lot of stuff, I'm sorry, but feel confused, it's a good state. Not your whole life, say for 2 weeks or so, and then it should start shifting again, okay? See you later.
Ja, hij is nog niet zo lang gedaan. Nee, maar we horen die groep te krijgen.
Do it. All right. Back.
Ja, bye.
Leave it on.
Dus als jullie iets voorbij zien, daar.
De deur wel gewoon dicht doen. Hè? Wat is dit nou weer? Irritant, hè? Staat hier gewoon een telefoon op te nemen nog.
Huh?
Ik ga hem gewoon stoppen hoor.